Today we are going to discuss about mutability data analysis and then get a hands-on practice on T-Bioinfo Server for the same. So what is mutability? Mutability in simple terms describes the observed rate of a given position to change or mutate. The mutability module generates a table in which each genomic position or a nucleotide is assigned with the most probable nucleotide variant. The variant is assigned based on the maximum of the low thresholds of confidence intervals, transformed to quasi-normal distribution for each alternative in the given position. So in the OmicsLogic Learn lesson on mutability, we discussed about mutation, SNPs and mutability. Now let's learn how we can use this information to analyze NGS data from different sources, locations, and years to derive biological meaning regarding the data. In the next steps, we are going to learn how we can perform mutability data analysis on NGS data using T-Bioinfo Server. We will also go through the complete pipeline for mutability analysis and explain it stepwise. Eventually, you will learn about the interpretation of the results as well. To build a mutability pipeline, choose Mutation Variant Analysis Pipeline under the Genomics section on the T-Bioinfo Server. Now let's set the parameters. For type of reference genome, it should be R genome GTF. Since here we are not using a model organism, we need to provide reference genome for desired organism, that is Plasmodium falciparum in our example. The format of NGS data needs to be FASTQ. Then we need to choose single or pair end reads. Here we are going to choose pair end, since in this example we have pair end read data. To upload the NGS data, the input data can be uploaded via using multiple options. Files can be uploaded as FTP or SVL links. They could also be uploaded as NCBI run table or as .txt files containing SVL or FTP links for all the samples. Next, we need to upload the reference genome of selected organism. Here we are using Plasmodium falciparum. Click on Add More option to create Get Link option. Subsequently, paste links for FASTA files of reference genome and GTF or GFF file for reference genome. Next, choose the groups. Drag the samples into respective groups. Here we are considering 2009 samples as control groups and 2015 samples as treatment groups. Now, let's start to build the pipeline. To begin with, click on the Start button. You will get a pop-up. Click on OK to start the pipeline. To align the raw sequence reads of NGS data, here we are using the Bauti algorithm. By clicking on the Bauti option, you will get a pop-up and click on Save to proceed further. Next, we need to perform per-nucle mutation analysis. Per-nucle is an algorithm that calculates the per-position coverage of the reference genome by all variants. The output of this module is a table with all the positions of the genome and the calculated coverages of each position by each variant. Next, we need to perform Bionorm 95. Mutation called Bionorm 95 is a module that performs calculation of the 95% confidence interval for frequency of every variant at the genome position based on Wilks' theorem for binomial distribution. To learn more about this module, you can refer to the Learn lesson on Genomics on OmicsLogic Learn Portal. Next, we need to perform mutability. The mutability module generates a table in which each genomic position or nucleotide is assigned to the most probable nucleotide variant. The variant is assigned based on the maximum of the low thresholds of confidence intervals for each alternative in the given position. The last step is to end the pipeline and give it a name. This pipeline can take more than 72 hours to complete or more than that, depending on the number of samples uploaded. Please keep patience when looking at the results. Okay, so now we have our results. In the mutability table, each genomic position or nucleotide is assigned to the most probable nucleotide variant, and the variant is assigned based on the maximum low thresholds of confidence intervals. Usually, this table is very large, sometimes difficult to handle on the PC systems. In order to understand and visualize whether the mutation or mutation frequency information can help us distinguish various classes of the samples in our data, we can use as an idea to understand and visualize the patterns within our data based on new features or principal components. Since the mutability output files are usually very large to handle, especially if you have large number of samples, that would be more than 20 samples, let's say, so it's better to not download the complete file. Instead, we can use the SVN link of the file. To do so, first we need to go to the download outputs of the mutability pipeline. Next, click on download job temporary files. Subsequently, need to click on SVLs, where you will get heterozygosity transform folder. In this folder, you will get all files.svl. Open this file in .txt format and copy the full link of the file named as heterozygosity log transform.txt. Now, to run PCA, go to the T-Bioinfo Server, click on the unsupervised analysis module under the data mining section. Although there are a number of options available to upload file on the server, since mutability output files are usually very large to handle, especially if you have large number of samples, it's better to use the SVL link of the file. Paste the link of the file in the blank box and run PCA. To run PCA, click on the Start button and now click on PCA-R library. Now end the pipeline, give it a name and run on cluster. In the output folder, you will get two important outputs. One is the PCA plot in the PDF format and second as a PCA result table. In the PCA table, we can see PC1, PC2 and PC3 that represents the percentage variance of the data. You can also draw the scatter plots based on these principal components using Excel. If you open another output, you will find the three PCA plots, that is PC1 versus PC2, PC1 versus PC3 and PC2 versus PC3, two of which are shown here. From the PCA plots based on the mutability results, we can clearly see samples from 2009 and 2015 from clear distinct groups. Further, one of the samples from 2015 looks like an outlier. This sample can be removed from downstream analysis. This analysis indicates strains of P. falciparum that can be separated from each other based on mutation frequency information. Thus, it would be interesting to know what are the important variants and mutations in P. falciparum between these 2 years. To learn more about the mutability analysis and how do you interpret the results, you can go to the course number 10 on mutability analysis and interpretation under Genomics section on OmicsLogic Learn Portal. For any questions, you can reach out to us directly at marketing@omicslogic.com and we'll be glad to assist you with your questions.